Okay, so I'm just updating my video on nucleic acids as the fourth and final macromolecule that I teach about. So in our organic molecules or macromolecules unit, we learn about carbohydrates, lipids, proteins, and nucleic acids. So in this video, we will focus on the structure of these molecules rather than a super in-depth discussion on their functions. So when we talk about nucleic acids, we're talking mainly about DNA and RNA, and as you read these descriptions, you can see how they both are involved in building proteins for our bodies. So DNA basically has our genes, which are the directions for building proteins, and then RNA helps with that process of protein synthesis. So their function is mainly to store and transmit our genetic or hereditary information from parents to offspring and guide protein synthesis throughout our cells. However, we have a whole nother unit on protein synthesis. So to see how these actually work will be a separate unit. And in this one, we're going to just discuss their structures and then compare and contrast them. So let's go ahead, though, and talk a little bit first about DNA. So DNA stands for deoxyribonucleic acid, and it is mainly the molecule that carries genetic instructions for life. So how does it do that, though, right? And so that's mainly going to be based on the sequence of 4 nitrogen bases. So you can see in the picture on the left, the colorful A's, T's, C's, and G's. Those are nitrogen bases, and that order or sequence matters. The order of the T, the A, the C, the G, etc., is what carries our genetic information from parents to offspring or from cell to cell. And so we will learn more about how that happens in protein synthesis. Now, the structure of DNA is a double helix. So we will see how it's made of 2 strands and forms like a twisted ladder structure. A lot of times, the structure of DNA is compared to a twisted ladder, and so we will make that analogy as we go. Now, this double helix structure is actually very helpful when it comes to DNA replication. It makes it easy to have identical copies of DNA passed from parents to offspring. But now my next question is, when we talk about these A, T, C's, and G's and how they're important for passing genetic information from parents to offspring, what are they, right? So DNA and RNA, but DNA is actually a polymer made of repeating monomers. So if I look at DNA, I know it has 2 strands, it's a double helix, and it's made of repeating units over and over again. So each of those individual units is called a nucleotide. So the monomer of nucleic acids are nucleotides. Now these nucleotides have 3 parts. So there is a negatively charged phosphate group. There's a 5-carbon sugar. In DNA, that sugar is called deoxyribose. And then there are nitrogen bases. Now, when we look at nucleotides, there are actually 4 different kinds of nitrogen bases. We have adenine, thymine, cytosine, and guanine. So when we look at nucleotides, though, here I have little cartoon shapes, but really they're made of a bunch of elements or atoms joined together into a nucleotide. So if I look here, I can see my phosphate group, my 5-carbon sugar, and the nitrogen base. So when we think back to macromolecules in life on Earth, while life on Earth is made from carbohydrates, lipids, proteins, and nucleic acids, those macromolecules are made from certain elements, right? So when we think about this, what elements do we see in these nucleotides? We see carbon, hydrogen, oxygen, nitrogen, and phosphorus. So nucleic acids have all 5 of the major elements that is necessary for life on Earth. Now, our next question then becomes, though, how are individual nucleotides joined together to form a polymer of DNA or RNA, right? But let's focus first on DNA. So we could join them into a single polymer like we learned in previous videos, how you join a monomer plus monomer plus monomer plus monomer will then form a polymer. However, DNA is double-stranded. So these nitrogen bases are going to need to pair up to form that second strand. Now, when we look at how this happens, we see that A will always pair with T and C will always pair with G. So in our first top nucleotide here, I have thymine, T. So if I were to bring in adenine, I see when I bring it in this way to form a pair, a nitrogen-based pair, I have the thymine is closest to the phosphate group, and that's not going to work for forming a double helix. So if we think about, well, what do we need to do to that adenine, to that A nucleotide? We actually need to flip it upside down. So in order to form a double helix, one side of that double helix actually runs in the opposite direction. So the two strands run in opposite directions of each other. Now, let's go ahead and talk a little bit about this structure of a double helix. So what I see here, I put red lines to help point out the covalent bonds that exist between sugars and phosphates. So between the phosphate group of one nucleotide, it is covalently bonded to the sugar of the next nucleotide. Now, these outer parts of the double helix are called the sugar phosphate backbone. So if I was climbing a ladder of DNA, my hands would be holding onto the sugar phosphate backbone of the double helix. Now, in the center, if I was climbing a ladder and stepping on things, what I'd be stepping on, the rungs of the ladder, would be the nitrogen bases. Now I see here again that A pairs with T and C pairs with G. Now I added little red bars in between. Those are going to represent hydrogen bonds. So the nitrogen bases are attracted and held together by hydrogen bonds between the base pairs. So I've used the word a few times, base pair. When you have two nitrogen bases form like a pair with hydrogen bonds between them, we call that a base pair. So A will always pair with T and C will always pair with G. Now, if you find a case where that's not happening, that is the case of a mutation. All right. So now, one reason why DNA is awesome is that if you know one side of the DNA, we can figure out the second strand because of base pairing rules. I just mentioned A always pairs with T and C always pairs with G. So here, my purple T, I can expect the base pair, the next nitrogen base, is going to be adenine. And then you can kind of quiz yourself as I go, right? So here we have the double helix. Now, we can explain this base pairing rules by saying that DNA is complementary. A will always pair with T, and C will always pair with G. If you separate DNA for one reason or another, you can always add nucleotides and build the complementary sequence following those base pairing rules. Now, this is important in replication because as we make copies of DNA, we're going to follow base pairing rules to make sure we get identical daughter strands. Now, because one side of the DNA does run in the opposite direction, we say that the two strands are anti-parallel. Okay. Now, we're almost done. So our second type of nucleic acid is actually RNA. And while we look at RNA here and compare it to DNA, I want you to think, you can hit pause in the video if you need to, but I want you to think about as many similarities or differences that you can see in these two pictures. Okay, so now let's go ahead though and compare and contrast both DNA and RNA. So if we look at the structure alone, we see that DNA is double-stranded and RNA is single-stranded. Then we see that the monomer, or the repeating building block over and over of these two polymers, are nucleotides. They're both made of nucleotides. They're both nucleic acids. Now, you kind of notice in my pictures that in the DNA, I made the sugar a dark blue, and then in RNA, I made it a light blue. And that's really to represent that they have different sugars. So while all nucleotides have a 5-carbon sugar, the actual sugar is different. In DNA, it's deoxyribose, and then in RNA, it's ribose. And that's where the DNA comes from and the RNA comes from in their names. And then if we look at the nitrogen bases, you'll see in DNA that it actually has a thymine, has T's, while RNA does not have T's or thymine. RNA has uracil instead. So this is more important later in protein synthesis units when you're learning how to build proteins from a DNA sequence. You want to be careful there that DNA has thymine while RNA has uracil. So to summarize again, our two nucleic acids are DNA and RNA, and they're essential when it comes to synthesizing our proteins and determining our traits and who we are. Now, again, I want to point out they're both made of nucleotides. They have CHONP. And that is my video on nucleic acids, and I hope it was helpful for you. Great job.